Hello and welcome to Urinary 1. This is the fifth and final podcast in Urinary 1 called the Renal Corpuscle. So if you listen to the previous podcasts, you'll know that the renal corpuscle consists of the glomerular capillaries shown in this cartoon and the glomerular capsule. So obviously the glomerular capillaries, that's where blood is flowing, and then the glomerular capsule is where urine flows. So it's really important to keep track of these two vessels uh, in the kidney, the blood vessel system and then the urinary tubule system. They're two different tubes in a sense, and they're carrying two different things. One carries blood, one carries urine. Also, in previous podcasts, we talked about three major renal processes. Glomerular filtration, when the plasma filters out of the glomerular capillaries into the glomerular capsule, which is the beginning of the tubule system. There's also tubular reabsorption when filtrate, so plasma that was filtered in the capsule is called filtrate. The filtrate gets in the tubule system, and then if the filtrate gets reabsorbed out of the tubules back into the blood, that's called tubular reabsorption. And if blood plasma that didn't get filtered into the glomerular capsule but continues to travel in the efferent arterial and the peritubular capillary, if that blood moves into the kidney tubule system, that's called tubular secretion. So now we're going to look at the renal corpuscle in more detail. So we're going to look at the cell types that make up the renal corpuscle. So in this cartoon, it shows the renal corpuscle in a little bit more detail. So that shows you a key. There's flow of blood in red, and flow of filtrate is the black arrow. So if you look at the cartoon, you see the afferent arterial that's bringing blood in. Upstream of the afferent arterial are a number of different blood vessels that we talked about in a previous podcast. But ultimately, the blood comes into the kidney via the renal artery, and then it branches until it gets to the afferent arterial, which the afferent arterial then brings blood into the renal corpuscle, and that blood then gets filtered. So the afferent, like I said, the afferent arterial brings blood into the renal corpuscle. When the afferent arterial is in the glomerular capsule, that afferent arterial then has become the glomerular capillaries. So in the renal corpuscle, like I said, there are the glomerular capillaries, and then there's the glomerular capsule. So what makes up the glomerular capillaries? The blood, obviously inside the glomerular capillaries are that's where the blood flows, and then the glomerular capillaries are lined on the inside by endothelial cells, but then sitting on top of the capillaries are these cells called podocytes. And podocytes, actually the cells called podocytes actually make up the visceral layer of the glomerular capsule. And then there's a space between the podocytes making up the visceral layer and the parietal layer of the glomerular capsule. So we've seen this visceral and parietal theme before. The visceral layer usually lays on top of the organ. In this case, the visceral layer is lying on top of the capillaries, and then the parietal layer usually lines the cavity or the structure that the organ, or in this case the capillaries, are in. And then there's usually a space between the visceral layer and the parietal layer. once blood is filtered or plasma is filtered out of the glomerular capillaries into the capsular space, it continues on into the kidney tubule system. It leaves the glomerular capsule and continues on in the proximal convoluted tubule. So the parietal layer then of the glomerular capsule is a simple squamous epithelium, and it doesn't have any role in filtration. The filtration of the plasma occurs through the endothelial cells of the capillaries and the podocytes of the visceral layer. And in the visceral layer, those podocytes, their histological designation is a simple squamous epithelium. But because that simple squamous epithelium is in the glomerular capsule, that simple squamous epithelium is called a podocyte. The capillaries themselves, so those are the endothelial cells lining the capillaries, are fenestrated capillaries. We talked about that in class, the different types of capillaries. Fenestrated capillaries just means that there's a lot of large intercellular clefts and large fenestrations going through the endothelial cells, so they're very porous. These capillaries are very leaky, and that's what you'd want if you're trying to filter all the blood in your body. The podocytes and the endothelial cells, because they're cells, they both need something to sit on, and cells always sit on a basement membrane. So the podocytes of the visceral layer, I don't know if you can see in the cartoons, but they've got sort of these projections, and those podocytes are called pedicels or feet, and those feet of the podocytes wrap around the endothelial cells of the glomerular capillaries. And then this interdigitation creates a filtration slit, and there's also a negative charge in the filtration slit. So it kind of makes a sieve. It's a molecular sieve, so that plasma that gets filtered out of the capillaries and into the glomerular capsule is passing through this filtration unit or sieve. So this is just another cartoon of the glomerular capillaries on the right hand side. So it shows the capillary endothelium, and there's lots of fenestrations. They're called filtration pores in this cartoon, but we've called them fenestrations before. And then superficial to the capillary endothelium are the podocytes. So you can see a large podocyte cell body, and then the projections coming off the podocyte cell body, those are called pedicels, so the feet of the podocyte cell. And then the feet interdigitate and sit on top of the endothelial cells, and this structure together creates that molecular sieve or the filtration membrane. Even though the endothelium of the glomerular capillaries is very leaky, the pores still are not big enough for red blood cells or big proteins to go through. So if you look at a urine sample and you analyze a urine sample, you shouldn't see red blood cells and proteins in that urine sample. The basement membrane that the capillary endothelial cells sit on and that the podocytes also sit on, it's a gel-like basement membrane. So it's got a negative charge. So if you're a positive smaller molecule, you'll be attracted to that basement membrane. But if you're a large negative protein floating in the plasma in the lumen of the glomerular capillaries, then you will be repelled by that negative charge, and you wouldn't filter out of the glomerular capillaries into the glomerular space.